Welcome to the Scientific Sense podcast, where we explore emerging ideas from science, policy, economics, and technology. My name is Gil Eapen. We talk with world's leading academics and experts about their recent research or general areas of topical interest. Scientific Sense is an unstructured conversation with no agenda or preparation. We cover a wide variety of domains where new discoveries are made and new technologies are developed on a daily basis. We are most interested in how new ideas affect society and help educate the world how to pursue a rewarding and enjoyable life rooted in science, logic, and information. We seek knowledge without boundaries or constraints and provide unedited content of conversations with researchers and leaders who love what they do. A companion blog to this podcast can be found at scientificsense.com, and this podcast is available on over a dozen platforms and directly at scientificsense.net. If you have suggestions for topics, guests, and other ideas, please send them to info@scientificsense.com, and I can be reached at gil@eapen.info. My guest today is Professor Hart, who is Professor of Neuroscience at Brown University. Her research focuses on the molecular and cellular mechanisms underlying neurological disease, sensory signaling, sleep, and fatigue. Welcome, Anne.
It's a pleasure to be here. Thanks for inviting me, Gil.
Yeah, thanks for doing that. So I want to start with one of your recent papers. Genetic modifiers ameliorate endocytic and neuromuscular defects in a model of spinal muscular atrophy. Before we get into the details, and what exactly is spinal muscular atrophy? Well, how does it present, and what are the symptoms?
Yeah, so spinal muscular atrophy is surprisingly common in the sense that 1 in 40 of us carries, in the white Caucasian population, we'll talk at least in the United States and most of Europe, carries a silent allele that has no impact probably on one of their chromosomes. But if you have a child with someone else who has a silent allele like this, the disease allele, then the odds are that 1 of the 4 children you have will have this disease. And the child is usually born seeming quite normal, but because they start losing the function, and then the neurons in the spinal cord that control your muscle movements die, these motor neurons, the child often will never sit up. It'll become unable to move its limbs. And before there was any treatment for this disease, the prognosis was really poor. Decades ago, it was like a couple of years. Now with better palliative and other assisted treatments and understanding how to keep the kids from getting infections, often these kids can live until their teenage years. But it's been exciting to watch the field find a way to treat the disease as well. So now there's some treatments that can really help people. That's SMA.
So you said 1 in 30.
1 in 40 probably.
1 in 40 of the Caucasian population has the defect. It doesn't materialize unless you have both copies.
Bad. Yeah. If the kid gets 2 bad copies, then there will almost always get the disease. And usually it's pretty severe. Sometimes there are families where it's less severe and symptoms might not show up until, the teenage years.
Yeah, but 1 in 40 is pretty high. And so we find it only in the Caucasian population, not else?
Sorry. Yes. It's a lower frequency in non-Caucasians, but it's still out there.
It's still out there. So 1 in 40, at least intuitively, and feels pretty high. So do we have sort of a genetic test that people can take to identify if they have the issue?
Good question too. Before there was a treatment for SMA,
Yeah.
because of the way, insurance and other things are set up and laws, there was not an easy way to test for SMA, and it wasn't, done for newborns or families. I think there are two reasons for this. One is there was no treatment, and so it wasn't clear what we could do. So testing the newborns wasn't going to change the outcome. Now there are pushes in multiple states to get testing done because now there is a treatment. So the other thing is it can be a little tough to test because let me think about it. How to explain it? More than one thing can go wrong with the disease gene. And so it's not like you have a magic, yes, no test here. It might be a little more complicated than that.
Mm. Okay. And it's a progressive disease, right? It gets worse and worse over time?
Yes, exactly. Most of the infants are born and they seem totally fine, but gradually they lose function of the neurons. And so then they lose ability to move their limbs, and eventually respiration becomes a problem too, just breathing.
Okay. And so you mentioned, so before we get into it, so you are using a model here and using C. elegans as the model?
Yeah. I can lean back and say when I started getting into science and I was interested in neuroscience back when I was in, high school and college, I didn't think about, I wanted to address questions about, how neurons, how does the nervous system develop and how does the brain work, what goes wrong in diseases. But I really never thought about, working with flies and worms because really those don't seem like they would have much influence, but all of this, that they would be similar. But it turns out almost all the animals on the planet have pretty much the same genes. Our neurons work pretty much the same way. And so one of the reasons people start using flies and worms is because you can ask questions fast and easy.
Yeah, so I guess evolution must be true then.
Yeah.
we have some evidence.
Yeah. So I did my training on using flies at UCLA, but then when I started training as a postdoc to do my own independent lab and set up my own lab, I moved to C. elegans, which is a little worm about the size of a period on a printed page. They're a millimeter long, but they have the same types of neurons, just in broad strokes that humans do. They have motor neurons that move their muscles. They have other neurons that tell the motor neurons what to do. They use the same signaling molecules.
Yeah, that's incredible. So really small. So how many? We can actually potentially count how many neurons.
Oh yeah, this was groundbreaking work done by John White and John Solston and others in the C. elegans field. The C. elegans, the ones we work with, have 302 neurons, and every worm has exactly the same ones. They're connected pretty much the same way to each other. So this neuron talks to this neuron, talks to that neuron. These neurons tell the muscles to move. So it's really convenient, you can imagine, instead of having to look at a mouse spinal cord or humans, hundreds of thousands of, millions of neurons, and every person is set up a little different. It's a lot easier to work on these things in worms. And I'm going to say, no one, animal research always has questions of how well we are treating the animals. There's less concern about that when you're working with worms and flies.
Right. They don't have a lobby yet.
Yeah, they don't seem to feel, they don't like nasty things that hurt them, but they don't have fear. We don't think they don't feel pain, so to speak. So one would imagine that the movements obviously is very critical for the C. elegans. So the muscular aspects of that, presumably it's a system that is really set up to move, right? In some ways. Yeah, it has to. The worms, flies, these little animals have to solve many of the same problems that large animals do, in the sense that you have to move to find food, you have to move to avoid noxious things, you know, you have to find a mate if you're going to mate. And so C. elegans has, you know, they swim along in water, or they can crawl on the ground, and I can just look at a worm and say, oh, that's a healthy worm that's moving really well. And another, if worms have mutations that make their neurons or their muscles work less well, and if those are severe, you know, I can look at that worm and say, oh, that's sad.
Yeah, so the 302 neurons, so can we engineer a defect into them?
Could you repeat that question? Sorry.
Can we engineer a defect?
Yeah. So we can do this a lot of ways. And over the course of the career, you know, our tools are just getting better and better. You've probably talked to people about CRISPR-Cas9 in your series, where we can go in and make the changes we want. So we can do that in C. elegans, and we take advantage of that in studying other diseases, and we'll probably take advantage of that again in studying spinal muscular atrophy. But at the time I did this work, that technology was not so advanced. And instead, what we can do is we can easily insert a chunk of DNA in the worm's genome, and that can change how the neurons function. Or we can express, you know, proteins using these. We can put proteins into the worm neurons that make them turn, you know, be more or less active when we turn lights off and on. It's really cool. We use what we call optogenetics to make the neurons more or less active.
And so SMA in humans, they have the neurons, but they're just defective. They're producing unexpected types of proteins. What exactly happens from mechanistically?
Sure. So mechanistically, when I talked initially about how you have the 1 in 40 of the Caucasian USA carry the disease gene, that means they have a broken copy that's defective of the SMA gene. And I'm going to talk about this sort of in broad terms. So the problem with the SMA is that this protein is gone. It's not made. And normally our cells, and especially our neurons, need this protein to move RNAs around and do the normal process of making lots of other proteins their cells need. So when they lose this protein, they start to, in effect, not being able to make the proteins they need to function. They start losing their connections to other neurons. They start pulling back their long, beautiful processes, and then they die.
Right. And so in humans, we find, you know, sort of the symptoms that they have difficulty moving, and it's a progressive disease. And so going back to the model, then. Is the paper about then we could modify these things in the C. elegans and actually see some beneficial effects?
Sure. There's a couple of threads that I think that would be of interest to folks running through this paper. One of them is that we can, based on work, you know, done by our friends and our collaborators elsewhere, we can, first of all, we can make mutations in the worm's SMA gene, let's call it. And the worms, if we make just a little perturbation in how the gene works, the worms look okay. And if we make a huge perturbation, the worms can't have babies, and those babies die really fast. That sounds familiar. So one of the threads in this is that if we want to study sort of these milder forms of disease that appear sometimes in human families, where the kids are born and are okay for about a decade, and then they get symptoms, we need a worm model that's milder. And so what we did was we put in a little perturbation into the worm SMA gene, and the worms look okay, but then when we make them exercise really hard, we can see they have problems, you know, keeping up with the normal worm. And so to make them work really hard, what we did was actually make their motor neurons using, we shine a light on the motor neurons, and that make the motor neuron work really hard. And then the worms got tired and they fatigued, and it took them a while to recover. So that was the difference in our mild SMA model between the normal worms and the SMA worms.
So they just have to put in more effort to get the same result.
Kind of. It's also kind of like, I could describe it as the normal worms could swim after we, gave them a serious stimulus. They could swim really fast and recover fast from all their thrashing around. Whereas the SMA worms, they started out swimming fine, but with the stimulus, they couldn't keep up, and then it took much longer to recover.
Okay. And what is the typical lifespan, C. elegans?
Really, it takes them about probably, depending on the temperature, a day to grow up from the egg to the adult, maybe 2, and then they'll die within a week or 2.
Okay. And so when you do the experiments, it would have an impact on the lifespan too, I would imagine.
Yeah, if we get rid, if the perturbation, if we lose enough of SMA function, the animals are going to die, just as they start to become adults. But or they'll have a shorter lifespan. Yes.
Okay. And so going back to the paper, so you're finding potentially interesting therapeutic targets through this work for humans?
Maybe. And so what I want to do is talk to you for a minute about how science is really collaborative. You've probably figured this out of everybody listening. And so when we're trying to think about what goes wrong in the neurons, why does losing this protein lead the neurons to die? We would read work from other people's, research. And a particular interest to me was work from Dr. Brunhilde Wirth in Germany. And she's a clinician and scientist who studies SMA families. And occasionally she would find a family where the little boy would have SMA, the son. And then she'd look at the genes of the sisters, and she would look at them and say, my goodness, this sister should have the same symptoms as the brother, but they don't. What is different? And she looked, because both the son and the daughter would have 2 bad copies of the SMA gene. So she started looking at other genes, saying, well, maybe they have some other gene that protects them. So she's done quite a bit of work over the years looking for protective genes in different families. So one of the protective genes she found was called, it's PLS3, that doesn't mean much, plastin-3. What it does is it helps the cytoarchitecture, the skeleton of the cell, helps the cells move. And daughters, the sisters who didn't have the disease, had more of this. So one of the things we're trying to do in this paper is figure out why more of this is helpful in the SMA.
Yeah. And so if you have more of PLS3, if PLS3 is overexpressed, it seemed to have sort of a protective effect. And could you do that artificially?
Yeah. First we could do that. We and others have tried to do this in different models. If you have if the worm or the human has has lost all of their SMA function, we've figured out that having more of this PLS3 doesn't help. But if there's still some function left, if it's a little mild, then PLS3 seems to help. It's tested. And we were able to show that in our worm paper here. The first step was saying, look, if we put more PLS3 in our sort of mild SMA model, the worms do better. And then we went on to try and figure out why they do better.
Yeah. And so going back to the humans, then that would imply, obviously this is early work. It has to be mild, or it has to be picked up early? If you pick it up early, would that be useful in this context?
I see. It would, if we could take one of these let's call them suppressors, one of these useful genes that Brunhilde's group is finding, and put it in someone else who has a mild case of SMA, then it would help. Or this is another possibility. We now have a treatment for SMA that partially helps. So in effect, it moves it from being severe SMA to mild SMA. So maybe all of those patients who are getting the current therapy could regain even more function in their life if we had this as an adjunct to that, as an addition to that.
Yeah. It's really fascinating. So you talked about 1 in 40 has the gene defect. In terms of actual incidence of SMA in the US population, what is the rate?
I think the math would suggest it would be about 1 out of 1,600 births. And I think it's a little less than that. I think it's about 1 in 2,000. But I'll say, as a faculty member, I'll have students in my lab or people who work in my lab and then go off to medical school and become doctors. One of them contacted me after they'd been in the clinic for, their first 6 months and said, by the way, do we have anything to help SMA? Because I'm seeing lots of these babies. And so it's pretty common if you spend a lot of time in, pediatrics.
Yeah, I mean, it's probably really difficult disease, especially because we don't really have at least a cure or a treatment that is really robust, right? So this will be really important. Yeah.
I think the cures that are out there they're getting better, but they're also pretty invasive. You want to inject into the spinal cord treatments. And I mean, it's at one level, it's a game changer. It's like, as a whole medical field, a therapeutic for a neurodegenerative disease that really, really works, but it's very expensive. The upside is, that we are making progress, though. I'm going to say, when I started working on SMA, I was surprised to learn how common it was. And then I realized, having an SMA baby, the baby is, it's a family tragedy. It's a personal tragedy, but it's not like ALS that so many people have heard of, because an ALS patient is usually also a young adult who knows a lot of other people, and they have a large social circle, and so many people, more people hear of it.
Yeah. Let's talk about ALS. You have another paper on ALS. Again, you use the elegans as a model here. Now, again, to set the context, what exactly is ALS and what are the symptoms?
So ALS is a disease. It's also known as Lou Gehrig's disease, named after a famous baseball player in the U.S. It's also what Stephen Hawking had over in Britain.
Right.
It's a disease that usually affects young to middle-aged adults, and it starts as a weakness, usually somewhere in like one of the limbs, and then it becomes progressive and spreads to the other limbs. And eventually, the patient can become incapacitated and unable to move their arms or their legs. And they're losing the same neurons, the same that are lost in SMA. But this is now happening in an adult, and it's a different genes are at fault. So there's been beautiful work over the last couple decades in the ALS human genetics field to identify first one, then a couple more, and then several more genes who, when there's a mutation, when there's a bad copy, a disease allele, the patient will probably get the disease.
And they're actually losing the
Yeah,
neurons.
the neurons start out probably not functioning well, but you have a lot of neurons that move every muscle in your body. There's not just one neuron per muscle.
Yeah.
And so, you start losing a few, it just, starts out being weak. But as the disease spreads, you lose more and more, you get more obvious weakness. And in fact, it can spread up, the disease can spread to also kill neurons up in the brain, which are also in charge of helping move the muscles, the motor neurons up in the head.
Right. And so it surfaces later in life. I would imagine Hawking is an unusual case, right, from a lifespan perspective. What do you
one of the
see?
frustrating and tough to understand things about ALS is that some patients will, the average prognosis is death within a couple of years. Some patients, though, will have a disease that progresses super slowly and they'll live decades, like Hawking.
And that is what happened to Hawking, right? Yeah. And so how long did he live from diagnosis?
He was a young adult, so he was probably, and he was, okay, you've caught me out here. I don't know. He must have been in his 60s, at least, when he died. I think Hawking is also another example of the specificity of this disease. It affects the neurons that help our muscles move. And for many patients, it doesn't affect how well your brain thinks, though, at all. For some patients, it does. But your brain is, in effect, doing just fine. You feel and you think just as well as you ever did. But it's just that you lose your muscle control.
Right. Yeah. And Hawking definitely thought, that's for sure. So going back to the C. elegans, so again, you're using the same model here. So what are we learning here from our models?
It's if we want to study a disease in mice or flies or worms, we have to try and find a way to, I'm not going to say you give the animals a disease, but that's kind of what it is. You have to create a model. And the simplest way that people started making models, especially because, there were only, some CRISPR wasn't available. Originally, when people started making models of ALS, they would often just take the human disease gene and put it into the mouse, the fly, the worm in lots of copies. And so there'll be a lot of disease proteins. And the animals would get sick really fast. And, that can be convenient because, you want to be able to test a lot of things. If it takes a year and a half for your animal to have symptoms, that's a long time to wait to do an experiment. So you can also learn, there's some things you definitely can learn from these disease models where lots of protein is made, and we've had lots of insights. But there are other things that, can be hidden in that model. It could be that, that's not because that's not exactly what's happening in the patient. The patient has a sort of more slow, a lower level of the toxic protein from the bad gene. And so with the discovery of the groundbreaking techniques of CRISPR-Cas9 genome editing, now we have the opportunity for this paper to precisely go into the worm genes and say, I want to put that little change that happens in the patient right there in the worm gene. It's exactly the same spot in the protein. And then I want to see what happens. So this paper was our first attempt to make what you could call a knock-in, a more precise, a non-overexpression model of ALS that might tell us more about why the neurons degenerate and die.
You talk about oxidative stress. What's the connection?
So the disease model we started with was the looking at one, there are many genes whose mutation can cause ALS. One of them is SOD1. People have been working on SOD1 for a long time. So SOD means superoxide dismutase. And I'm not going to go dive deep into that, but it's one of the proteins that helps the cell deal with reactive oxygen species. Oxygen is good until you have too much of it or you change it around a little, and then it's actually dangerous, right? So free oxygen radicals are dangerous. And your cell has to deal with those, and that's what the SOD1 protein should normally be spending its time doing. But when the disease allele comes into play, we think now, based on the work that's coming out of our lab and others, that you have two things happen. One is the cell loses its ability to deal with this superoxide free radical oxygen stuff because there's less of the normal function, and the disease allele, the change that the disease makes in the SOD1 protein also makes that protein toxic or dangerous in some other way to the cell. So two bad things from one change in a protein.
Yeah, so this free oxygen radicals, I mean, it could have lot of issues, right? Not just on the neurons, but generally on the body, I would imagine. So do we see sort of other peripheral issues in ALS?
Yeah, no. You've touched upon one of the big questions in the neurodegeneration field, especially the ALS field or SMA, in that these proteins are expressed in every cell of your body, but for some reason, neurons and motor neurons, in fact, are exquisitely sensitive to what goes wrong. And so your skin cells and your liver cells and your bone cells are mostly okay. It's somehow your neurons, specific neurons, that can't deal with it. There are some neurons that aren't affected at all. So I would say that there's pretty good evidence that the metabolism, your ability, is a little bit different in maybe an ALS patient, especially as disease progresses. They tend to lose weight faster on the same diet, maybe. Yeah. But the overwhelming and huge thing that goes wrong is the neurons selectively.
And so if I understand this correctly, and so diminishing levels of SOD1 reduces the body's ability to essentially deal with this bad stuff, this free oxygen radicals. And when the level of those things are high, they seem to disproportionately affect the neurons, and ultimately over time it ends up in the issue that we are talking about, right?
Definitely one of the theories about one of the contributors to ALS that's caused by mutation of SOD1 is that it seems like the cells aren't able to handle these free radical oxygens, these superoxides, as well as a normal cell. And the question becomes, is that all there is to it, or is there more? And if we have two types of neurons dying, the spinal cord neurons and the neurons in the brain, they're actually two somewhat different types of neurons. Are they both equally sensitive to this, superoxide oxygen paraquat stress, or is one of them more sensitive than the other?
Right. Can we therapeutically sort of add stuff so that the body has the ability to get rid of the free radicals?
Getting that into the neurons would be tough. Yes.
But so it's not necessarily a systemic effect.
I see where you're going. Sorry. If you, there's a delicate balance, right, in our cells between they have to make deal with, we use oxygen, do superoxides all the time, that because we have, we're oxygen based for our mitochondria. If we can't shift it too much because that runs us into other problems. The other cells of our body aren't really affected so much by the SOD1 mutations. I'd be a little nervous about trying to change your whole global overall and how you handle oxygen. We'd rather to just try to help the neurons.
Right. Yeah. This is always a problem, right? You try to do something, and all the effects of that in the complex system in other parts of the system we can really forecast, and that ultimately leads to
it's a problem for
problems
every therapeutic approach people want to make. Usually, even some of our best drugs, right, usually have off target or other consequences.
Yeah, I think, and I don't know too much about that. I think this issue exists even for stuff like vitamin C and things like that.
Oh, yeah.
When people overdo
don't take too much vitamin A. You'd think it'd be good for you, but no. Right.
Yeah, it's sort of the same issue. So where are you heading? Where are you heading with this? With C. elegans, you now have a method by which you can very finely engineer the disease into C. elegans. We can observe them, and we can try different approaches on them. What are the things that you're working on to progress?
We are working on a couple things. So, in fact, this first paper was a test case saying, oh, look, we can engineer models that look much more precise into C. elegans. So we've gone ahead and done that for several other genes and created disease models for them. And now we can do a couple things with all these models. First of all, we can try out, one of the overarching questions is, first of all, why do neurons die? And do the neurons die differently in an SOD1 ALS worm or patient than they die in a ALS caused by some other gene like FUS or HNRNPA1? So even though we call ALS one big disease, that's, we sort of lump everyone together, it may be that it's really more than one disease. And so we want to understand that. Yeah. So we also can use our worm models, and we can have millions of worms at a time, and we can look for mutations in other genes that might help the neurons. So I'm going to call these suppressor genes. Because we can have so many worms, we can look for suppressor genes in worms, figure out how they work, and then hand them off to our collaborators who work on flies or mice, and have them test those genes in the flies or the mice. And then if something works in worms and flies and mice, odds are pretty good it's going to work in humans. It's going to be relevant. So we have a lot of collaborative projects where we are either taking suppressor genes we're identifying now in worms and finding them and then moving them to our friends to test in other animals, or genes that they think help there, and we're testing them in the worms, and then we're trying to figure out, like, mechanistically, why the neurons die as well. There's lots to do. Well,
job security.
Well, I mean, admittedly, job security and figuring out the basic mechanisms is fine. It would be lovely, though, to figure out an effective, something that could be a therapeutic, and then I'd like to have us be able to fix, help the patients, cure them, and then we can all focus on some other disease.
Right. Yeah. So SMA ALS, it looks like a lot of common features there. Neurodegenerative gene defects. What do we know about the environmental effects? Are there features from the environment that sort
yeah.
of combine with genetic defects?
It's for SMA, my short answer is probably not. For ALS, that's much more of a question in the sense that if you start reading, websites or older books or older studies, people would talk about sporadic ALS that just appeared in a person and no one else in the family had the disease, versus familial ALS, where you suddenly realize, oh, that's what the grandmother had, that's what the father had, and now the kids are probably at risk. That's familial. So we used to think that most ALS was sporadic because we couldn't find a gene. But now that we know more genes, more and more we realize there's a big genetic component to ALS. But there's still a large section of patients where one can't find one of these genes. And so that means, and now I'm speculating, that either there are a lot of really rare genes. Each family is like a unique sort of, we very rarely does this cause disease, or something about the environment that person's genetic makeup and they were in led to ALS. And I don't want, you can now think through your head of all the things that are bad for neurons and think maybe those combined with a specific like ALS mutation might lead to disease. And people are testing that sort of stuff.
Yeah, so do we see changes in incidence rates over time and across the world?
I mean, it's a little more common in males than females, like many diseases, but not enough to make you, shocked. It seems like it might be of a little higher incidence in the military and in athletes or people who are super athletic. But again, it's not overwhelming that. So it could be just that there's something about ALS, maybe this is also speculation. Maybe there's something about someone who's going to develop ALS that makes them, more athletic. It works really well in the beginning, and then there's a crash that we call ALS. So maybe they're more prone to go into athletics because they succeeded on average more. I don't know.
Yeah, you mentioned SMA being more frequent in Caucasian populations. Is that the same for ALS too?
Not as much, but the genes that cause, that we know about already, that cause ALS in different. Like I think the Japanese, if you study ALS genetics in Japan, some gene mutations are more common than other mutations in the United States.
Right. And for ALS, obviously, you have to control for age. Different countries have different percentages of age populations.
That's true too.
Right?
It's another question also, and I'm circling just slightly back to your conversation, but ask a question about environment. Does environment change when symptoms show up?
Yeah.
It could. We don't know.
Yeah, that is kind of curious about this. I think you called it spontaneous ALS or something like that. That's sort of cute, right? So the conditions are there. It doesn't really surface, and then suddenly one day.
If you ask most of us why it takes so long to show up, we would probably say the biggest factor in that is that these disease mutations, the bad toxic protein is slowly accumulating. And on top of that, you're slowly losing, because of that, neuron function. And eventually you reach a tipping point where things start going to hell in a handbasket.
Yeah, a lot of the diseases ultimately gets there. And I think, it's sort of a plumbing problem. It's a system designed for maybe 30 years. Now we're taking it to 75. It's not really designed for that long.
It's like the worrisome thing. How many of us will get Alzheimer's or a dementia if we all find a way to live to,
Yeah.
100.
Right. Right. Yeah. So in conclusion, and yeah, I know that you're doing a lot of work in this area. So if you look forward 5 years, where do you think, where is your sort of highest level of optimism in terms of therapy for both SMA and ALS? What type of therapy do you think we will get to? And whether, those things can ultimately cure this or just
Oh,
slow it down?
SMA,
What
I
do you
think
think?
SMA is on a good track in the sense of it's clear what we need to put back into the neurons, what gene, and they're going to get better, I think, and less invasive slash expensive for the therapy. And then for the SMA patients, it'll then be a question of how much better, what else can we do, what else can we add? So SMA is on a pretty good glide path to not, if you get treatment early, not be fatal, and to be
Yeah.
something where decades of life with good mobility should be expected. ALS is trickier. If you 5 years from now, it's possible that we'll be able to slow. I think in 5 years we'll have treatments for some patients, especially if they have a gene we recognize, where you come in to lab, or if it runs in your family. Within 5 to 10 years, we'll be able to say, we're watching
Yeah.
you, we're worried about this. Maybe we can talk about this sort of treatment. But um, the screening, the societal questions are just as big, in the sense of how easy is it going to be, and how much do people want to invest as a society in us, for you could have a mutation in any of these 30 genes.
Yeah. 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 I mean, that is the sort of the overhanging question for
Yeah.
healthcare in general, right? It seems like technology is developing a lot faster than the regulatory framework, or in some sense, a
Appetite.
societal um, uh, acceptance, so to speak, right? And so it's a really complex thing. But a good thing is
Yeah,
technology
exactly.
is really
Sounds like you should
advancing
have
quite
if you
fast.
haven't already, have another podcast on that topic of, you know,
Yeah, yeah, definitely look into that.
it's a pleasure
Excellent.
talking
Yeah,
with
this
you.
has been
Take
great,
care.
Dan. Thanks so much for spending time.
Bye.
Okay, you too. Bye.
This is a Scientific Sense podcast providing unscripted conversations with leading academics and researchers on a variety of topics. If you'd like to sponsor this podcast, please reach out to info@scientificsense.com.